So Anjali Henders is the project manager at SALSA, based at IMB, and she's also working very closely with Paul Talman. Thanks very much.
Thank you. Well, I have received the button. I'm the last runner, so we will head for home as fast as we can. So what I really want to do today is firstly I would like to thank the organising committee for inviting me to present this project on behalf of Professor Naomi Ray, who's based here at UQ, and also Professor Ian Blair at Macquarie University, who are the lead PIs on this project. So just a little bit of background. I don't need to tell you too much about the Ice Bucket Challenge, but 2 years ago, many people around the world doused themselves in a bucket of cold water, and this raised huge amounts of money specifically for M&D research, raised awareness, and here in Australia, where we don't like to be left out, we single-handedly raised $3 million for research based in this country. So $1 million was then awarded by M&D RIA for a very specific project, a 3-year project that was to lead a group of Australian researchers to really try and understand and make some inroads into the causes, the genetic influences, and any kind of research-based project for M&D. So in May 2015, Naomi and we were in China working on a collaboration with a hospital in China for MND when we received a phone call from Janet Nash to say that we were successful, the SALSA project was successful in being the recipient of this $1 million grant. Here are some of our QBI researchers participating in the Ice Bucket Challenge a couple of years ago. So last year, this time exactly last year, all the jacarandas were out, and we were very fortunate to have a guest speaker at this inaugural event, Nancy Freites, and she was very inspiring, and it was a lovely opportunity to meet her and really talk to her about the initiative that they had started in her family in America and how it spread across the world and made it to the bottom of the planet, as she referred to us. She had just spent a long time on the plane, and she was genuinely very excited that a bunch of clinicians and researchers, again, beyond the equator, which was sort of this new concept for her, was going to make significant inroads and really try and attempt to make some understanding of MND. So who are we? We are a very large group of researchers all around Australia. We're comprised of clinicians, clinical research nurses and coordinators within the clinical teams, some established groups that were already doing very successful M&D research at Macquarie and in Western Australia. And then we also have the new team that joined this consortium, which was based here in Brisbane at the University of Queensland, which brings the expertise of genetics and genomics to this very complex disease in trying to disentangle the components of the genetic influences and any environmental influences. So these are just the sites where SALSA is currently participating, and the ones with the stars also have a joint laboratory. There is a very large biological collection component as part of this project, and those samples will be sent to those sites for the purposes of sample receipt and processing and storing those in -80 freezers for use in this project. So why the SALSA project? And the S on the front of ALS does stand for sporadic. And as we know, there's about 90% of the cases that are presenting at clinic come with unknown cause or with unknown causes. Those people who fall on the 5 to 10% where there's one or two other members in their family that also have ALS are being diagnosed as having a familial ALS or MND diagnosis. So it has been a long journey to get to the point where we are actually today, and this... Wrong button. Sorry. It started back in 1992. We are many years on from that, and just recently in 2016 we've been able to add to this list of known genes that have an influence in ALS. We've added another 4, and that paper came out just recently in in August. So even though we do have a lot more information about the genes that are involved in ALS, you can see that there is still many, many cases where people are presenting at clinic that don't carry these genes, and that is the search, and that is one of the major aims of SALSA, is to try and look for those. So what are our aims? We're investigating the sporadic nature of an onset of ALS, and to do this, because we know that MND is a rare disease, we need as many teams as possible and as many clinics as possible to come on board and recruit people into our projects under very standardised protocols. So some of the slides that Paul showed you earlier regarding the parts of the body where, or the sites of onset and the classification of the disease becomes very, very important. And although each of the sites has already been doing this, we wanted to create a standardised platform to make sure that at the time that the data is put together, there are no questions, there are no concerns about integrating that data effectively and being able to use it completely. We've also So the part of that process was to get all of the clinicians together. That has been somewhat of a challenge, and I think we've all agreed now on a set of very clear measures that we will be collecting as part of the Salsa Protocol. The second part of the data that we will be collecting comes from the biological sample. And so at this stage, we are just collecting a whole blood sample. I will talk about that in a moment as well. But the data and the different types of genomic data that we're going to be able to get from that sample will allow us not only to share the data within Australia, so nationally, but also will, with other collaborations that we are working with overseas, we can put all of this data together to make it a much larger data set, and with that we have real hope then of actually finding any genetic influences. So what exactly is the Salsa Protocol? And again, Paul has referred to some of these standardised bits of information that we will be collecting. So we have information around onset. This is the time that the symptoms first appeared, the time that a person first went to any medical professional about their symptoms, when an actual diagnosis was made by a neurologist. All of these are now very, very standardised and gives us absolute timeframes for the onset of this information, because we know that different people are presenting in different ways. We have different regions are affected, and again, Paul alluded to that, and we're testing the reflexes as well. The functional rating scale is a standardised questionnaire that is administered in most clinics, both here in Australia and overseas, and it gives us an assessment of how the patient is functioning in their everyday life. And so as the disease progresses, their functioning ability will actually change during that course. And that is very important for us to track, not only from a clinical perspective, but also in any changes that are occurring physiologically, we may be able to actually trap that or look at that and really set time points that will match any of the genetic information that we're collecting. Ancestry is also becoming very important, and asking people what their ancestry background. That's always difficult for someone like me. I'm your classic moggy, so I answer all the boxes, and so I'm an outlier. But when we are putting genetic data together, it is very important to have an understanding of the origins of that sample, so that any differences that we do see between the different cohorts where we've merged the data together is actually only due to the clinical information or the disease information, and not actually due to kind of cultural differences or background ethnic differences in terms of where the data has actually been collected. So this was the biological sample that I was referring to. So at the moment we're just collecting a whole blood sample. It is about 40 mils, and we are planning to collect this sample every 3 months for as long as someone is able to provide that. And from that sample we are collecting a range of different components. So plasma and serum, so when the sample comes in we do spin that down and we collect off the different components. And plasma and serum are collected and stored to look for the biomarkers. So we store these in -80 freezers. It preserves and ensures the validity of that sample when we go to use that, but we can pull that out at a much later time and actually investigate something if it comes up in any of our results. We collect blood in EDTA tubes, and the buffy coat, or the cell layer that comes off that, is what we use for DNA. And the DNA is what we use for many of our genomic assays that we're looking for. And so we use that to look for mutations or changes and differences, not just between people who are affected and people who aren't, but also between cases, because we know this is a very heterogeneous and complex disease, and there are many differences between patients that we also are looking for and trying to understand the cause. So one of the tools that we've had to develop, and I say the royal we, but I actually mean our database developer, Hasti, has spent a lot of time trying to provide efficient research tools for each of the clinics to be able to collect this data in a standardised way, in an efficient way that doesn't impede the flow of the clinic or the clinical appointments. These clinics can become very, very busy and very, very crowded, and so we didn't want any part of the protocol to be missed. And so what we have spent is time working on this particular portal, which every site around Australia will actually have access to. Now this also has a public page component, which we will be, so that members of the public can go on and Google this site. There is information about the SALSA Consortium in the researcher section. This will be updated constantly to include other research projects that are happening within the sites and maybe very specific to that particular site. And of course, if anybody has any questions about their participation in the project, those will be where all of our contact details will be. So because we are a large team and we span a very big physical mass, one of the ways to train effectively was to bring everybody here to Brisbane, and we have been training here at UQ for the last 2 days. It's been quite an intensive day, but I think it's been extremely rewarding for everybody involved. And we've gone through the entire protocol for SALSA. And one of the things that is very important is making sure that we conduct this type of research, particularly with its focus on genomics, under all of the ethical requirements that are put on us, but also the legal requirements that we have to fulfil as well. We have to work under the Australian Privacy Principle and the National Data Sharing Policy and the National Human Tissue Storage Policy, as well as making sure that anything that we do will also comply internationally, so that we are also able to share that data and work collaboratively to further the research. So this is the team. We've had a great 2 days. We've had lab tours. And I think everybody has come away from that understanding what the protocol is all about and what we absolutely need to do to make sure that this project is successful and is done to a quality standard. What's next? We're constantly reviewing the set of clinical measurements that we will be using in this project. And that means that we have to stay in touch with what's happening in the clinic so that we remain current and clinically relevant, not only within Australia but also internationally as well. Technology is constantly evolving, and we're looking at new ways that we can squeeze out any extra bit of information from that biological sample that we can to use for this project. And as part of our online portal, we'll also be adding other data collection types, including environmental measures, adding imaging and neuropsych. And so that's me. I just want to thank all the partners involved. We've had a great training session over the last 2 days. And if you have any specific questions, I'll be around at lunchtime. Thank you.